Anyways, okay, thanks. Yeah, thank you to the organizers. I will start my presentation. So today I want to tell you a bit about chemical reaction network-based framework for the origins of biochemical life. And this is some work that I did as a part of my PhD thesis with David Baum at University of Wisconsin. And so for people who are maybe unfamiliar with the question, the origins of life question sort of phrased in the following way. So we know we have, the Big Bang, sort of 13.8 billion years ago, and then we have the early formation of the early Earth, some 4.5 billion years ago. And from around 3.7 billion years ago, we have a fossil evidence of the first form of life. And the question is sort of, for the origins of life, sort of what happened in between. And the and the fossil evidence has, the life form that existed, I mean, is kind of similar in the way that it also had DNA, and as we have now. So some questions, and I mean, of course, there's many ways of asking these questions, but the questions that I'm interested in, or I was interested in for my thesis, was were sort of more conceptual rather than practical. So it's like so they were along the lines of, is there a mathematical framework in which we can talk about chemistry and biology somewhat on similar terms? So, like, what would it mean for a chemical system to evolve, for example? And in a more sort of concrete way, the question that I would like to formulate and ask is, can complexity reliably emerge and transition to phases with even higher complexity? Because, for example, the DNA has it's composed of really long polymers. So how did it happen that from an early Earth where you have only small molecules, you have phases with longer and longer molecules, something which is a precondition for something like the DNA to come about? So these are the things that I would be sort of interested in talking about and formalizing. So, this talk will not be a very mathematical talk. It'll be more, I mean, there is of course maths involved, but the point of this is sort of to build some sort of conceptual framework in which we can pose some of the questions that maybe people in origins of life are interested, but more mathematically. So I'll tell you a little, I mean, I will probably not tell you anything about CRNs because Eugenie has already done a great job. And then I will tell you this framework for evolution of complexity in CRNs, and I will also give some concrete models. In these models, I will point out the role of autocatalysis, and since this is the autocatalysis seminar. And finally, I'll end with connections to evolutionary biology. So, yeah, just so you know, this work is, it's all it can be found in this archive preprint and has been recently accepted by PRE. So, yeah, it'll be published soon. Um, okay. So, yeah, I don't think I should spend any time explaining to you what chemical reaction networks are. But essentially, the high level picture is that, these are directed multi-hypergraphs. So with two types of vertices and two types of edges. So you have a vertex for species, a vertex for complex, and then you have edges for stoichiometry, and then you have these directed edges for reactions. But it's, yeah, uh, some examples maybe are better to talk about. So the reason why we we sort of use chemical reaction networks as a framework to talk about this is because they're used, you can use them to talk about nuclear fission, so something at the nuclear level, at a metabolism level, and also at an ecological level. So in an earlier paper sort of, and I mean, this is kind of well known that you can also get all sorts of ecological behaviors in chemical reaction networks. So it really is a very versatile framework in which we can talk about dependencies and sort of relationships at all different levels of hierarchy of organization. Right. So now I will present to you the framework and some concrete models. So what exactly is the framework to do? It is to ascertain if a stochastic chemical reaction network is capable of evolution. So I will define all of these things, of course. But sort of here's the outline. We'll start from a CRN. We'll obtain this thing that I call an attractor transition graph. Then we'll obtain a complexity graph. Then we'll superimpose the two to get an evolution graph. And then from the evolution graph, I will give specific criteria whether or not that CRN is capable of evolution. So yeah, okay. So we can talk more about the implications of this towards the end. Um, so step one, start from a complete thermodynamically consistent CRN. And by all of those terms, what I mean, and something also Eugenia mentioned, is that we start with a rate constant assignment for a CRN, says that your system has a detailed balance attractor, and says that it's a detailed balance system. And we know that there's this sort of correspondence between detailed balance systems and sort of this thermodynamically specified system where each species comes with chemical potential and and so on and so forth. So so let's say we start with a CRN where now you have listed all of the species, whether they're internal to the system that you want to call or that you want to investigate, whether or not it's evolving, or external. So, there are species in the environment with which it's interacting with. We have all of that information, and we sort of decompose our CRN into this tripartite sort of, you make three partitions of this. And we have certain species that appear only in the environment. Then there are some certain external species that are interacting with certain internal species through reactions in the boundary. So this is what we call boundary CRN. And then there's this thing called that we call internal CRN. Terrible terminology, but you have to stick to it once you say it. Is that this is the CRN that composes only of the internal species of your system. And the reason why I use internal, and external is kind of very silly. Like I wanted to distinguish it from an internal external partition. So this is not that, so there's three, and why I couldn't call it internal boundary external, I don't know. But yeah, but I thought that would somehow add more confusion. So just to make things more clear, we have this redundancy. But anyway, so this is just the setup. Let's see a concrete model, and we'll be using this throughout the talk. So CRN for polymerization in one monomer. So let's say your species are a monomer, dimer, and then trimer, and so on, and enmer. And this line at the top sort of indicates the that it is like I with a line on top. It means it's an imer. And the polymerization reactions, let's just for simplicity assume end-to-end addition. So, 1 + 1 goes to 2. I mean, reacts, and everything is, of course, bidirectional. 1 + 1 reacts to 2, 1 + 2 reacts to 3, and so on and so forth. And then we have, again, some kind of interaction with the external environment. So we have these things called inflow and outflow reactions. So Y0 can convert into a monomer. And then we have a catalyzing reaction, whereas a catalyst of size C, it takes something from it reacts with Y sub C to make to again give you back C plus a monomer plus YC plus 1. And for sort of what the way I will sort of develop this further, we're going to chemostat these Y species, these external species, and and so what we then get is this simplified, inflow and outflow reactions where you have sort of environment going to 1 and C goes to C plus 1. And as a very concrete example, let's just fix our polymerization scheme to polymers of size 4 and let the catalyst be of size 2, right? So this is like a very concrete example that we'll be working with. And back to the sort of diagram of this tripartite, partitioning that I showed in the beginning of a CRN. This is what it will look like. So in our boundary, we have the monomer and the dimer. The monomer is inflowing and outflowing with this Y0, and the dimer is catalyzing, using the environment, the formation of new monomers. And then in the internal CRN, we just have the polymerization scheme, so like end addition. And okay, so this is sort of the setup. This is a concrete example of a CRN that we'll start with. But remember, like the point of this talk is essentially to give you a framework. I mean, the example itself is not as important as some of the applications of this that could potentially come out. The next thing is we obtain an attractor transition graph. So let me quickly review a few things. I mean, again, Eugenia just went over this, so I really don't need to do this. But of course I will not do it. I will not show you the math. I'll just give you the sort of the brief outline. Is corresponding to, let's say, a CRN, which is shown here on your left. You have you can have many choices of kinetics that this CRN can instantiate. And what this will give you is this will give you a flow field on on the velocity of the population of the chemical. So there's a one species system, you have only chemical X. And so under a certain choice of rate constants, this is what the velocity field will look like. Where now the interesting sort of thing here is that this velocity field crosses zero, 3 times. So these are called fixed points. So this system has 3 fixed points, and 2 of these fixed points are attractors, and one of them is a repeller. So if you start in the neighborhood of the attractor, you end up your system. If you start sort of here, your system will relax into the attractor. And this is what will happen in this sort of deterministic system. So this is what we mean by an attractor. And one of the things is that if you take a completely deterministic system, you're sort of losing out the fact that actually chemical reactions fire stochastically. And that is, the way you model that is the number of so what reaction happens next is a function of what is the population right now, and each reaction will happen with some probability. And if you just wait long enough time and then you plot the histogram of how much time your system has spent in each point, then this distribution will converge to the stationary distribution. Well, if your system is not growing and such. And the thing is, the stationary distributions might look, sort of, it would seem that each system, each stochastic instantiation will have a different stationary distribution or something, maybe, but like you might naively expect that because it's stochastic. But indeed, if you sort of descale the improbability, you descale the stationary distribution, like say take the log of it and you scale it by some appropriate quantity, you get something that you can analytically or numerically find, and this is what is called as the non-equilibrium potential. And this is sort of unique to you can you I mean you can for a given system with rate constants, you can find a non-equilibrium potential. And this is typically hard, but the to find analytically for more than one dimensions. But we have an algorithm to do exactly this action functional gradient descent algorithm that I have with Eric, and this is on PRE. And so essentially the point being here that we have also developed, I mean, this was also part of my thesis, to develop a technique to actually numerically estimate or find to whatever degree of accuracy you want what the non-equilibrium potential should be, or the log of the stationary distribution. So now the attractor transition graph, let me first give an example and then we can look at the definition. So let's say you have a system where the mass action kinetics is this magenta line below, and the non-equilibrium potential looks like the blue line on top. So what it's saying, so for the attractor transition graph, we make vertices which correspond to the attractors, and you draw solid edges between neighboring attractors. So an attractor here is like, it neighbors the attractor over here. And so that's how you have edges between A1, A2, and A2 and A3. And then you have edges towards the more stable attractor. And by stable, I mean the one that has a lower non-equilibrium potential. So in this case, clearly the first attractor has the lowest non-equilibrium potential, and the third one has the highest one. But you can also look at an example where the first one has the highest non-equilibrium potential and the third one has the lowest one. And then your attractor transition graph will sort of look like this. So you go from A1 to A2 and A2 to A3. And so this is what we mean by an attractor transition graph. The point of sort of doing this is that if you now think that you have a stochastic system, and let's say if you look at the figure on the right, and you start close to the origin, your system will first relax into attractor A1. But since the probability of attractor A2 is more than the probability of attractor A1 in the stationary distribution, under some and under some conditions, you can say that, I mean, and the condition just being that, the number of particles on the system is really not infinite. You'll say that eventually your system will fluctuate out of that attractor A1 and make its way into A2. It kind of must, because the probability of the system spending time in A2 has to be higher, right? And then eventually the system will fluctuate out of A2, and it's more probable that it ends up in A3. And once it settles there, that's somehow the most stable attractor that it can be in. And of course, there will be some fluctuations, but they will be like just exponentially less likely to get out of A3 than to get into A3 once it finds this most stable attractor. So that's what this attractor transition graph is supposed to sort of tell us. It's supposed to tell us if you're looking at a stochastic instantiation of this CRN, what will the dynamics look like? Okay. Now our model, this concrete model that we're sort of investigating, you under some choice of rate constants, it can have two different attractors. So you have an attractor, and where the average length of your polymer is 1.1 in this lower one and 2.4 in the higher one. And let's call the lower one as A1 and the higher one as A2. And we'll see that, I mean, for the same case, the non-equilibrium potential is at A1 is actually higher than the non-equilibrium potential at A2. So from what I've just told you, the attractor transition graph of this system will look like A1 goes to A, like, there'll be an edge that points from A1 into A2, okay? Where A2 being the attractor with the higher average polymer length than A1. Right. So that's the second part where you obtain an attractor transition graph. Then we obtain, so now I need to tell you a bit about complexity graph. So complexity of species and attractors. First, let's just take a step back. Useful abstraction for chemistry that people have, like Christoph and others, and people in the graph rewriting community have been using for, yeah, since before I was born, is um, is that molecules can be seen as graphs, and then reactions are then graph rewrites. So so essentially, you have when you have a reaction like H2 plus O2 gives you H2 or something like that, you can see that as two different graphs coming together to create a new graph of H2O. And reaction mechanisms are graph rewrite rules. So for example, aldolization or any of the basic mechanisms in chemistry that you might have studied, you can formalize them as graph rewriting rules. And so the idea is that we can use methods from algorithmic complexity to sort of, to assign a complexity to species. And one sort of naive way in which we can do this is, let's say we specify the CRN using these graph rewriting rules, right? So we have this sort of a graph grammar that generates our CRN. And then we say the complexity of a species S is sort of found in this sort of network expansion way. So we say, let's fix some species to be the food set. These are just available from the environment. And then the minimum number of reactions that you need to produce a species S from the food set, that can be the complexity of a species. Now, there are, of course, other ways of defining complexity as well. One of them is the assembly index, but it's a very similar logic. So assembly index is the minimum number of joining operations necessary to construct the species from its basic parts. So this is a definition that you can find somewhere in either this article or the ones that follow it. But the idea is again similar, right? So you're interpreting chemical species as graphs, and then you have certain graph joining operations that you would then assign that and the number of these operations, or the minimum number of these operations, is then what you will call as the assembly index of that species. Now, for the polymer model, both of these cases will be the same. And let me just, remind you real quickly again. Let's say the food set is these external species Y0 and YC. And so it takes exactly one step to make a monomer, right, from the environment. And then it'll take 2 steps to make a dimer, 3 steps to make a trimer, and so on and so forth. So this would be, uh, so in this way, you can say that, the complexity of a species of a polymer will just be how many monomers does this polymer have. And you can also see this in an assembly index setup. If you might be thinking, oh, but like if you allow for more complicated additions, which is not just end addition, yeah, uh, things will should not. Yeah, I mean, all of those assumptions or any of those choices can be made, and they will have different repercussions. But what I'm providing here is more like a framework for like conceptual outline. But now let's say you have your favorite choice of complexity of species. What then we can do is we can assign a complexity to attractors. And remember that attractors are populations. So what we need a way of taking a complexity of species and getting a complexity for a whole population. You can either define like a total complexity, we can define the average complexity, where Q is the concentration of a species. So Q sub S is the concentration of species S. Again, you can also look at assembly is one of these things where it's weighted average complexity, where the weight is like exponential of the assembly index. So what I'm saying here is it's it's compatible with the formulation of assembly theory. But once you have, all of that, we get what we call a complexity graph. So let's say, in this sort of mock system, you have different attractors, different polymer attractors, and let's say you have attractor A1, A2, and A3. What we would say is that to get a complexity graph, you use dashed edges between neighboring attractors, and the direction is towards the more complex attractor. So clearly A3, the average length is higher. So from the notion of complexity that I just introduced in the previous slide, A3 will be more complex than A2, which is more complex than A1. And with the particular model that we were working with, where we have only 2 attractors, our complexity graph is simpler. It just goes from A1 to A2. Okay. So now finally, what we will do is we will now impose these 2 graphs to get an evolution graph. So there's no definition needed there. But then I will now give you criteria for saying when does a stochastic CRN, for which now you have obtained an evolution graph, what is the criteria for evolution and what type of evolution is it? So if your evolution graphs looks like this, so you have A1 and A2 is more complex and more probable in the stationary distribution. So that's what the solid edge of the attractor transition graph shows. Then this would be what we could call directional evolution. And if you have something like, if you have some kind of pitchfork, that A1 can go to A2, or A1 can go through A3, and both A2 and A3 are both more complex and more probable in the stationary distribution, then this is what we would call a historically contingent evolution. And then if you have chains of this, like, if they're infinite chains, either in one direction or in multiple directions, then this is what we would call open-ended evolution. So let's just say, so back to our running example. Step one, we start with a CRN. We saw that the attractor transition graph looked like, A1 going to A2. The complexity graph had the same sort of flavor to it. So this would be so then we would say this model the stochastic model is capable of directionally evolving from attractor A1 to attractor A2. And again, in the origins of life setting, just recall that if you start very close to just having monomers, eventually your system will relax into these lower attractor, and it will relax into A1. But just by the stochasticity in the system, it is sort of bound to fluctuate out of that attractor and escape into the and relax into the attractor with the higher polymer length or the higher complexity, right? And these are the kinds of models that we're interested in the origins of life setting. So, I mean, I will say more about that later, but let's just see another example where instead of just working with monomers, if I was working with two types of monomers, so you have a 1 comma 0 and 0 comma 1. Again, you can see the details in the paper, but we can find a system where now your attractors look like this. So you have one attractor here, another attractor where the concentration of, let's say, monomer of the type 10 is high, and then another attractor where the concentration of the type 01 is high. And indeed, we can have the rate constant such that that the evolution graph of this looks as it would for a historically contingent evolution. So we can find CRNs that are capable of historically contingent evolution as well. And more importantly, this is how we can formalize a notion of historically contingent evolution in biology for CRNs, right? Which is sort of the purpose of this. Now, let me quickly sort of remark on the role of autocatalysis here before going to the conclusion. So, so the choice of catalyst that I had, where C1 was my catalyst, we have two types of reactions. We have this reaction, all of this polymerization scheme, which takes you from monomers by adding a bunch of monomers to C1. But since C1 can also go to C1 plus 1, so C1 can bring a monomer into the system, this is what is an example of a autocatalytic subnetwork. And just these two reactions, what I've shown, this is a minimally autocatalytic subnetwork. It's in 1 and C1. And it is not non-ambiguous or unambiguous, but this is fine as we formulated in this paper here. Now, a feature of this model is that assigning more of these species, so more catalysts, can increase the number of attractors in a one-to-one correspondence. And just so to remind you of some terminology that we had in the beginning, C1 is what we would call a boundary species because it actually takes in from the environment, and everything else, and like, I mean, these are all internal species, but essentially, and I make this sort of more clear in the preprint that you can look at, which we don't have time to go over now. But you need some of these molecules, or you need some of these species to be able to pull in resources from the environment, right? So that you can climb up to higher complexity. And this is what the role of autocatalysis sort of does in our models, is that you have some of these species which will pull in more from the environment to produce more of themselves. And in this way, the system will sort of climb up in complexity. And just to sort of make this more clear, by adding more species, more of these functional species, we can add any number of attractors. And indeed, if you add like an infinite number of these catalysts, you can have open-ended evolution. But you cannot, of course, get infinitely many attractors for free. Now you will have infinitely many parameters to play around with to be able to get this. But indeed, there is a big space where your system will be capable of evolution, and it's not like measure zero or something. And more details of this for this particular model can be found in the paper. So this was the criteria, and now I've shown you how to get sort of these basic minimal models which can show directional evolution, historically contingent evolution, or open-ended evolution. So I'll just end with a few connections to evolutionary biology. So as we know, that in life as we have it, sort of formation of membranes in which CRNs can be instantiated is sort of key. And one thing here that I would like you to sort of consider is suppose you have a membrane which has a multi-stationary CRN, and it has one of these CRNs which, as we've shown before, can show multiple attractors, and it can show this and is capable of historically contingent evolution. So let's say the complexity sort of goes higher as you come up. So red is more complex than blue, and also you can see the non-equilibrium potential of L2 and R2 is like lower than the non-equilibrium potential at the origin. So if this is sort of instantiated in a membrane setup, what you might sort of see is that your system starts out with O, but then it sort of some of these membranes, they fluctuate, they find themselves in L1 and eventually in L2, and some of them find themselves in R1 and R2. And then each time division happens, you might have like mutation events where now if L2 is dividing and it, let's say, divides somewhere close to the origin, well, there is some probability of it going to the origin, of it relaxing into the origin attractor, and then some of it goes to R1, right? And in this way, we can sort of formalize the notion of mutation. We can also formalize the notion of heredity, in the sense that maybe things which when you have a membrane in L2 divide, it it one of them is still in L1, or maybe one of them is in L2. And you can see that the core autocatalytic species, whose concentrations you need to be high at to maintain the system in attractor L2 or L1, you know, when you're dividing, the concentrations in the two division cells is also high, so it somehow retains its memory of what it was before the division. So I mean, these are all sort of very vague, and I'm going through this very quickly because I'm at the end of my presentation time. But the questions that I'm sort of interested in is, what are some key features of life that can or cannot be found in stochastic CRNs? And from sort of what I've shown, at least naively, it would seem that you can indeed formulate, heredity, mutation, and this notion of evolution that's uh, uh, that's important for evolutionary biologists to even think. You can formulate all of this in the setup of stochastic CRNs in a sort of with very little problems, with a very few sort of concepts. The only concept that you need to borrow out of your stochastic chemical reaction network theory is this notion of complexity, but which has a deeper rooting into another aspect of CRNs, which is this graph rewriting aspect. So really, CRN theory is sort of broad and rich enough that maybe we can sort of bridge this gap between what chemistry is and what early biology was. And that was sort of the point of this investigation. So acknowledgements. This, as I was mentioning, this was work done during my thesis, and my advisors were Eric Smith and David Baum. And this was done when I was at University of Wisconsin-Madison. Yep. And thanks to everyone who helped along the way. Thank you.
Thank you very much, Praful, for this wrapping in some sense and wrapping up and pushing forward.